Welcome to EASL Studio, your weekly hepatology broadcast news. In today's episode, we are discussing on the topic of understanding sex disparities in metabolic dysfunction-associated steatotic liver disease, which I'm sure from now on we will abbreviate as MASLD. Last Saturday, it was International Women's Day, and this year's theme being Accelerate Action. And to my opinion, this is desperately needed because the Worldwide Forum predicts that with the current actions and speed, we will reach gender parity only in 2158, which is roughly 5 generations from now. I like that EASL strives to reach gender equality in all its activities, although I would say that with today's faculty, we're not doing very well in that respect. But today we are discussing the biological determinants of gender differences that impact on liver health and more specifically on MASLD. My name is Saskia van Meel and I'm not an expert on this topic at all. But with me are Munifer Demir, based in Berlin, and Sarah Della Torre, based in Milan. So I hope to learn more on this interesting topic today. So now the first question. So you always read that most clinical trials have been done in male subjects and patients. How is that in hepatology? And is this a problem?
Yeah, I'd like to take the first question if I'm allowed. And I think, unfortunately, we have this situation as well in hepatology. There's no difference to the general situation. And this comes because of concerns about hormonal variability, like menstrual cycles and the difficulties for interpreting the data, clinical data coming from that. And on the other hand, most importantly, the potential to get pregnant. And this, due to this systematically excludes women, especially in the childbearing age, or a majority of women in the childbearing age, so that we don't have a variety of age distribution and also a minority of female participants in the clinical trials.
Mm-hmm. And how is this in preclinical trials, Sarah?
It's similar in basic science too. In general, males are used as a reference model in several fields of research, and is similar also for the pathology field. So the majority of studies are performed on males and also on male cells, for example. So the most used cells G2 cells are from males, for example. And we have very few studies where females, so animal female models are included, and very few studies where males and females are properly compared. This is another point, because in some cases females are included, and this is the same also for clinical trials, but there is not a proper comparison. So sex is not considered like a variable or a co-variable, and so put all together, males and females, is not something good. Is actually not the way to do proper sex-based studies.
Yeah, I'd like to add on that. The problem resulting from them is that we know that we have sex-specific treatment responses and also sex-specific natural history for MASLD, for example, but we are not able to address them and understand the causes or the reasons for these differences. And for that, I'm very happy that we have this EASL Studio today to get more awareness in the field that we have to do something in this respect.
Yeah, Because if the female and male livers were the same, then there was maybe not a problem. But you say there is a problem, but they are very different. So, and if I then ask the both of you, how are these female livers then different from male livers?
I start first. Okay. First of all, we have a smaller liver for women. In female, it's about 20% smaller than the male livers, which comes with a lower capacity for metabolism, drug metabolism, for example. And this is shown very nicely that for this acetaminophen, where the capacity to metabolize is 30% lower and slower. And with this slower metabolism, you have a longer toxicity to the liver. And this is one of the anatomic differences, but we have also differences in, for example, in alcohol metabolism. We have lower gastric levels of ADH, alcohol dehydrogenase, and for that we have a higher alcohol toxicity, which is reflected by the fact that we have lower thresholds for alcohol in women to be toxic for the liver, for example.
Sarah,
yeah,
are there
sure.
more differences that you can?
Yeah, the factors actually that can lead to sex differences in the liver are very a lot, because some of them depend on sex chromosomes, because the liver can express some female or male specific genes that are X or Y linked. There are some epigenetic factors also. The pattern of growth hormone is pretty different in males and females, and this is responsible for big differences in gene expression. So if we change growth hormone pattern in males, we can reprogram the liver metabolism toward a female-like phenotype.
So how is this pattern different of the growth hormone in males and females?
Yeah, because in general in males it's more there are several peaks, and they can go up and down, and in females they're more continuous but higher. And in experiment where males were treated with GH, with growth hormone, some growth hormone dependent genes become expressed as in females. So there is this possibility also to reprogram the liver. Obviously, several other factors, also sex hormones obviously have an effect in the liver, because estrogens for females and androgen for males may regulate the gene expression, but also protein expression and the regulation of metabolism and inflammation in general. So we know that from clinical studies, but especially from studies performed with specific animal models, for example, castrated females or males, so varectomized females. So, for example, females without estrogens that are a model of menopause, for example, or in models where the receptors for sex hormones, so estrogen receptor or androgen receptor, are not expressed. So in this case, we can see significant changes in the regulation of gene expression and of liver metabolism, and on the susceptibility also to MASLD in diet-induced experiment, for example.
Yeah.
Yeah.
Yeah, very interesting, fantastic. To add on that, I think in these estrogen receptor knockout mice, we've seen higher levels of fibrosis, right? Yeah, and I think there's also evidence for that testosterone from the other side promotes lipogenesis and also HSC activation. And on the other hand, the estrogen, at least to my knowledge, suppresses HSC activation, right?
Yeah, because it can regulate the inflammation. In general, estrogen are able to resolve the inflammation also by regulating the polarization of macrophages or Kupffer cells, which are the macrophages resident in the liver. So this kind of regulation of inflammation may prevent the activation of stellate cells and so of fibrosis. But I know that from a clinical in clinical studies, there are some kind of counteracting results, because in some cases, women seems to be protected against steatohepatitis or fibrosis. In other cases, when they develop fibrosis, this kind of fibrosis is more severe than in males. So there is a kind that means that X female-related factors and estrogen too can protect females from this pathology and from the progression of this pathology. But when this pathology start and progress, maybe in some cases can be worse in women than in males. So the point is that we should consider both sexes because the prevalence and the incidence of MASLD is different, but also probably the pathways that are relevant in males and females are different. So we cannot study only one sex and think that, okay, for the other is the same. This is not possible. We need to study both sexes and probably try to also think to different approach for females.
Absolutely, Sarah. I think what you said is also supported by the clinical data we have and the prevalence data we have. On the other hand, in younger ages of women, we have this potentially protective effect of estrogen, so the prevalence of MASLD is lower. But with a menopause, the prevalence gets equal to men, but the severity of disease, like getting fibrosis and progressing to cirrhosis, is higher than in women. So supports absolutely what you already said from the basic science perspective. Yeah.
Yeah, the point that for women, so MASLD can develop later in life because of the menopause, but for women that means that there is an aging effect and also a reproductive aging effect. So we have both, and the point is that in addition to estrogen, also other factors such as genetic factors, some kind of mutation or epigenetic factor may influence in a different way the development and progression of MASLD. For example, women that express some specific mutations, for example, for PNPLA3 gene, may have a different risk of developing MASLD according also to the reproductive ages. So according to the presence or absence of estrogen, so to premenopausal or postmenopausal status. So we should consider also that sex hormone effects may interact in a different way with genetic risk factors for MASLD.
Yeah, definitely. And on the other hand, we have also this societal and socioeconomic factors affecting women differently than men, like having, for example, access to healthcare or getting the right diagnosis at the right point, being monitored equally, and so on. And I think we have also differences from this point, which comes more from the society point. Yeah.
So you mean that women make less use of hospital care?
I think when we think about Germany, Italy, or Netherlands, I think it's not a problem, and probably they are those who are more compliant, go more to doctors. But I think when we see it more globally, I think that women have, and I think we have evidence for that, that they are a lower access to health, to the health system than men, when we see it in a global perspective.
Okay.
Yeah, this is a gender effect, I think, which is more related to cultural factors.
Yeah. And we discussed already that well, a lot comes from the Y chromosome or the extra X chromosome, and that's probably a lot of hormone influences. But are there also other things? Sarah, you already mentioned epigenetic status. Can you give some examples of that? And how is this epigenetic status different?
So it could be some kind of epigenetic factors may influence. We don't know so much about, but probably can be also in some cases can be also related to sex hormones, because you should think that sex hormone can also program the gene expression or regulate gene expression according to the level of sex hormones, and this can be also related to the fact that also in liver we can see also a sexual differentiation that is due to sex hormones, and especially to estrogens, because some differences are obviously related to the biological sex. So I mean XX, XY, but they need to go through the proper developmental stages, which are associated to specific window of the life, and one is around the birth, where is a kind of organization of the also liver gene expression, which is due to estrogens in males, and then an activation effects which is around puberty, so after puberty. So a way to program the organism to be able to respond to sex hormones in a proper way and to sustain reproduction. But obviously to sustain reproduction, also the metabolism, and also the metabolism of the liver, which is very important for nutrient regulation, for the metabolism of nutrients is important.
And would you say that the sex disparity is especially relevant in MASLD, or is it for all liver disease?
No, it's a general topic. I think in liver disease, we don't see this disparity only in MASLD, for example. Autoimmune liver diseases are more prevalent, and women are more susceptible for autoimmune reactions. But even in this situation, besides PBC, which is mostly done in women because 90% of affected people are women, but for example, in autoimmune hepatitis, it's not well studied what the sex-specific responses to therapies, for example, are, or the susceptibilities, although we know that.
Okay. Yeah. So yeah, what do you think is the current state of art for MASLD? So what do we know now?
We should try to improve our way to perform both basic research studies and clinical studies, so clinical trials, by including more females. And in the basic research, we should try to include females, but also try to use the proper models. So if we think that estrogens can be relevant for a particular mechanism that is involved in MASLD, we should try to dissect and study better this mechanism by using, for example, female mice with and without estrogens, for example, or try to use mouse models that do not express estrogen receptor, or for males, try to use mice that don't express androgen receptor or cannot have androgens or the ability to produce testosterone or DHT, which are two completely, they may have different effects also in males. So we need to use properly this kind of models and try to understand how sex hormones and changes in sex hormones during the lifespan can affect MASLD. And for the clinical trials, I can pass the question.
Yeah, but that will establish what the effect is of the hormones, right? But what if it's not all about the hormones? Because the Y and the X chromosome, they have a lot more genes, right?
Yeah, sure. But so it's just a way. So the point is try to select the proper condition. So if we know that a sex chromosome may have an effect, okay, we can compare males and females. To understand if sex hormones may have an effect, we can try to use castrated mice and compare with normal mice. If we think that the effects are related to GH, we can try to understand if changes in growth hormone levels may have an effect. So the point is try to think to all the potential factors that may influence sex differences in the liver and try to set up these conditions. So try to use the proper diet, try to select males according to sex, but also to sex hormone levels. Try to think also, for example, to other factors. So circadian rhythm may have an effect, microbiome may have an effect. So obviously we cannot do everything, but if we select the conditions the experimental condition, especially for animal models, because it's easy probably then in clinical trials. Could be the best way to try to properly collect the data and try then to analyze in a proper way.
Yeah, and this is more for the basic science, right? To understand how males and female livers are different. But if you now think of more translational work, because there are already a lot of pre and clinical trials on drugs for MASLD, how do you think those experiments should be redesigned to address also this sex disparity problem?
Yeah, the most important thing I think is that we analyze the data specifically for women and for men, and to see whether the outcomes are the same in the both groups. And it's assumable that they will not be the same. And furthermore, to go beyond that is to find out what's the reason. Why does it work better in women, or does work better in men? And on the other hand, we have to go more to like precision and sex based approaches, like doing analysis on, or doing studies on treatment responses, like doing, for example, studies to test sex-specific therapies, for example. Probably we need different doses for women of the same substance. For example, I just take an example. We take GLP-1s, and probably women at this age need more or less because they have some other metabolism and another microbiome and so on. And I think that's the most important thing, where we have to go there. And for that, we need a kind of scientific policy in clinical trials as well as in basic science. We have to make clear that this is not like an option to study for sex-based responses or outcomes, but we have to do that. To perform a valid and meaningful study, you have to also look for outcomes in sex-specific outcomes and therapy responses. I think that would be the way.
Yeah. And you both now already mentioned the microbiome. So, and the microbiome will determine what the liver sees in terms of nutrients and other things. So how is the microbiome different in males and females? Are there really differences, sex differences there?
From those studies which were performed in humans, we have signs that there's a different composition. And more importantly, I think we have higher levels of LPS. Therefore, we have evidence in males than in females, and LPS is very important in the gut-liver axis and triggering the immune responses then in the liver via TLR4 and so on. But I think there's a huge lack for specific analysis, I would say. I'm coming from the field as well, and our numbers usually are too small to do meaningful statistics in a sex-specific manner for composition, but also for the metabolites and so on. But what we have in this regard shows that there are differences in the composition and also in the metabolites.
Do you know anything about the preclinical models, Sarah?
I remember a very good paper published some years ago where people worked with germ-free mice. In these mice, they see a reduced number of sex differences in the liver. So without a proper microbiome, the differences in the liver are less. Sex differences in the liver are less. That means that because I know probably in humans is too much complicated because too many factors, the diet, lifestyle, obesity, so inflammation and other factors may influence the microbiota population and so microbiome. So it's quite complicated to find some kind of conclusion from clinical trials. But for this kind of experiment in mice, we can understand that also the microbiome can influence sex difference in the liver. And we know that males and females for mice at least, differ for the microbiome, and also the sex hormones may influence the microbiome. So there is a reciprocal regulation of microbiome and sex differences, and is related once again to sex hormones.
Yeah, and we have differences probably due to the sex hormones in the bile acid metabolism, for example, which highly influences the microbiome composition, but also the gut-liver axis in general. So definitely, I would agree on that. But again, our numbers in the human studies, at least, are too small to do meaningful statistics. So we have to really focus on that and go there, that this is not an option just for looking for sex-specific differences. This should be standard of science or standard of clinical trials.
Yeah, really interesting. And well, I've learned a lot. That is really an important topic. And I'm wondering, how can EASL help the field, or is there a role for EASL to make this better for future clinical and preclinical research? What would you suggest to EASL?
First of all, policy, I think. We should have a EASL policy on how to conduct clinical trials and how to conduct preclinical trials with regard to sex-specific differences and to address them. This would be one thing. Creating awareness and bringing the field more to this to this sensibility for this topic, but also to the importance for scientific outcomes. That would be one thing. And another thing would be to help us to share our data so that we can use, for example, AI models to do that what we haven't done before, but we can do it now. We should put our data together and analyze all the specific liver diseases for sex-specific differences with the help, for example, of AI tools.
Okay, very interesting. And Sarah, do you have anything to add on this?
But policy is important. We know that some years ago, the NIH mandated the researcher to increase the number of studies that included women or female animal model in research, in clinical trials and in basic research. So there are very few years from that, so 10 years, so it's not so much for research, but actually we can see probably now has started a small increases in studies that included females. The point is not limit the studies to inclusion, but try to perform better the studies, try to compare better the results, and try to report better the results obtained in the proper way. So actually this chart is already a way to try to increase the awareness on sex disparities on liver and on MASLD. We should probably try to increase the knowledge, try to put together as tonight people that work in the field, also people that have different skills and different expertise from basic and clinical research, try to increase this kind of actions, because we need before make people, make researchers, make clinicians to understand that these differences are relevant, and we know too few about them. So probably we know something that is like a small part of an iceberg. So we need to increase this kind of research, but we need to convince people that this kind of studies and the proper approach is important. So this could be a way we should increase this kind of actions.
Yeah. Yeah, and for that we need, I guess, to show the problem.
Yeah.
And that, I guess with both your studies, both in the basic science and the clinical science, I think we are getting to that. And if we show that the problem is huge, then I guess also the actions will follow, I hope. But I do hear you that, you both think that there's a role for EASL. So let's see if we can make them do something in this respect. They probably will involve you, so.
Yeah, probably, yes.
Yeah.
So,
would be fantastic.
Yeah. So I guess we are nearing the end. Are there any final points you want to make, Sarah?
Actually, I would say that probably we can also try to think that we have different type of female populations. So as in basic research, we can try to study different type of females according to, for example, sex hormone levels and so on. Probably also in clinical trials, we should include more women, but different populations. So in general, women are less than men in clinical trials. We should like double the number of women, because we have a subpopulation which is protected from MASLD. This is our women in the fertile age, more or less, okay, if we think to normal people without obesity and so on. And another kind of population, which are the old women after postmenopause, because you know, in some cases we suffer from the lack of knowledge from clinical trials. So we can try to study some stuff by doing basic experimental studies, but we have this lack of knowledge from clinical trials. And we need to try to have a better exchange, but we need I can understand is a big challenge, a big issue, because for humans and clinical trials is not so easy, and the experiment are the studies are very long, and the point is that it should be useful also to have longitudinal studies. This is something that we don't have at all. We can do that, for example, in animal models. We can try to follow mice for several times and see how MASLD progression can change. We don't have this kind of, I think, of data from clinical trials. And this could be useful, because probably the progression of the pathology is completely different in men and women. So it could be fine to try to program and set up also longitudinal clinical studies.
Yeah, importantly, not only having the data longitudinal, but also having the samples longitudinal so that we can look for the markers we detect from the preclinical models, for example, validate them, and then understand the disease and do kind of translation in both directions, like reverse translation from observations we see in the clinical situation, but also translate findings from the preclinical models to the human situation. But therefore, we don't need only the data longitudinally, we also need the samples for that.
Yeah, sure.
Ah, great. Well, I thought this was a great session. I would like to thank you very much for this. And I want to announce the next episode, because in the next episode of the EASL Studio, we will explore ways to reduce alcohol-related harm. And remember to become a member and join the EASL family. Thank you very much. Have a nice evening.
You too.
Thank you. Bye.
Bye-bye.